Hi, I got another really an excellent question. I've gotten a bunch of really good questions, but I really like this question. I'm going to try to answer it. It's actually a series of questions, and it has to do with lactate and MCT transporters, so monocarboxylate transporters. And the first kind of overarching question is, I got it here in front of me, is what is the difference between lactic acid and lactate, which is being removed and utilized in the muscle? So I think that the two things aren't the same. I think lactic acid is in itself kind of like a false term, and it's kind of used by it was kind of developed this idea of lactate being responsible for acidosis. And that's obviously not the case. Lactate has nothing to do with proton accumulation, hydrogen ion accumulation. And so I referred to a paper by Robert. It's called The Biochemistry of Exercise-Induced Metabolic Acidosis. And if you read through that paper, he clearly identifies ATP hydrolysis, the breakdown of ATP to ADP, which generates inorganic phosphate and protons released. So basically, you generate acid by breaking down ATP. And if you break down ATP very rapidly and you can't recycle all these protons, then you generate an acidosis. What Robert also goes on to say, which is really interesting, is that lactate actually stops acidosis. So lactate absorbs or hooks up to these protons. And that's why it looks like lactic acid, right? Because it's taking up these protons. But if you didn't have lactate, you would have more protons around, essentially, because the lactate wouldn't be buffering. So lactate is a buffer of acidosis. So you have diseases like McArdle's disease, which have a diminished performance, which could also be a reason because of this inability to have lactate as a buffer, with other reasons, of course, as well. And what lactate actually is just basically like a byproduct of glycolysis. It's basically sugar further down the glycolysis track that you can keep using once space capacity of mitochondria is available to oxidize this lactate, right? So generally, lactate has three major features or like components of what it does. So that first, it's the most important one for exercise science, is that it's the major source of mitochondrial respiration. So like 80% of lactate produced is just oxidized again somewhere in the cells. Then it's a major glycogenic precursor. So lactate is important in generating more glucose or glycogen. And then the third is it acts as a signaling molecule. But the first two, the major factors in kind of the way that energy is moving in the muscle. The second question is is MCT1s. So do MCT1 transporters take lactate from the blood to be used in the mitochondria? So it's like an importer of lactate. And followed up with, do MCT4 transporters remove lactate out of the muscle, so like an exporter? And that is generally the way that we look at these two, MCT1, MCT4 transporters, which are the main ones kind of relevant to exercise. Is MCT1s are importers of lactate and MCT4s are exporters of lactate. And you can see this in that MCT1s are much more prevalent in red fibers and highly oxidative fibers, muscle fibers, and MCT4s are much more prevalent in white fibers, so low oxidative or high glycolytic fibers. So that makes sense. That's kind of the way that these function. With a key thing here with the kind of lactate discussion and the lactic acid discussion is the MCT4 fibers. Then the MCT4 transporters in the highly glycolytic fibers, where you're generating a lot of lactate and you're taking apart a lot of ATP to produce energy, which means you're generating a lot of protons, a lot of hydrogen ions, a lot of acidity. Having the ability to move a lot of lactate out of the cell, out of those glycolytic cells to maintain pH, becomes very important because lactate becomes a buffer, right? It's helping you move out. So you need the lactate production and you need the MCT4 capacity to move this acidity out of the cell. which is one of the reasons that you actually, so both MCT1 and MCT4s can be affected by training. So endurance training increases both. And generally, if you look at the literature, what it said is more that in order to maintain MCT4 levels, you need to do intensity training. So with MCT1 more related to endurance training, MCT4 a little bit more related to high intensity training. But which would again make sense with the fiber type distribution of where you find the two. But it's not super clear. So again, the literature, there's some back and forth in this. There's literature that shows no changes in MCT1 or MCT4 with exercise training. There are ones that show differences. There are ones that only show differences in MCT1, but not in MCT4. So it's certainly not super clear how this is going to work, because the last question that was asked is there a function of what type of training have you seen to improve the concentration of these transporters? So I don't measure any of these transporters, so I can't give any answers of what I've done. And in the literature, like I said, there's some contradictions there. But I would say the general statement to make is endurance training is MCT1 and high intensity training for MCT4 would be my suggestion. I think that covers all the questions. Great questions. I hope it helps the answers. These answers help, and thank you very much. Bye.